Hello, it's Jihad Ali from Grand Canyon University, Concept of Human Physiology 2, supervised by Dr. John Tyson. And today we're going to talk about the function of pulmonary surfactant as a part of the respiratory presentation. So without further ado, let's get started. Pulmonary surfactant is composed primarily of phospholipid and key protein. The lipid constitute about 90% of the composition of pulmonary surfactant, while protein constitute about 10%. Lipid compose 80 to 90% of its molecular weight, of which the most abundant species are phosphatidylcholine, phosphatidylglycerol, and phosphatidyllinositol. Specifically, phosphatidylcholine constitutes approximately 70% of the lipid portion of surfactant, and it exists as a relatively unique form known as DBBC, which stands for diplamidophosphatidylcholine. Together with surfactant protein, DBBC provide the surface activity of surfactant. The remaining types of lipid, including phosphatidylserine and phosphatidylthanolamine and sphygmomyelin, appear to be present in a relatively small amount. This lipid composition is well conserved among vertebrates. Surfactant contains 4 associated proteins: surfactant protein A, surfactant protein B, surfactant protein C, and surfactant protein D. 2 of these proteins, A and D, are hydrophilic, and the other are hydrophobic. Surfactant protein A and surfactant protein D are members of the family of innate immune system proteins, termed collectins. These proteins have in common an terminal nitrogen link region and a C-terminal lectin domain that bind carbohydrates in a calcium-dependent manner. Binding sites for these lectin domains are found on bacteria and viral surfaces, and this in part is responsible for the role collectins play in innate and adaptive immunity. The hydrophobic surfactants protein B and C are stored and secreted along with surfactant phospholipids. Surfactants B is an indispensable protein that play a role in enhancing the surface tensions, reducing probabilities of surfactants, and also appear to have some antimicrobial activity. The role of surfactant C, one of the most hydrophobic peptides known, is uncertain, but its high degree of conservation among species suggests an integral function. Let's talk about surfactant life cycle synthesis, secretion, and recycling. Surfactant is synthesized in the endoplasmic reticulum of alveolar type 2 cells and transported to the Golgi. For further modification, most of surfactant components are stored in the lamellar bodies, as we see on the pictures over here, until they are secreted into liquid hypophase on the alveoli by exocytosis. Surfactants form a lattice-like structures called tubular myelin, as we see in step 6 on the picture, which is transported after that to the air-liquid interface to form a monolayer of surfactant film. The phospholipids are either internalized and degraded by macrophage, or recycled back to the type 2 cells for reuse. Surfactant proteins A and surfactant protein B and surfactant protein D are also synthesized in club cells and internal bronchioles. Pulmonary surfactant is a mixture of lipid and protein which is secreted into the alveolar space by epithelial type 2 cells. The main function of surfactant is to lower the surface tension at the air-liquid interface within the alveoli of the lung. In order to understand the function of pulmonary surfactants, we have to understand the alveolar surface tension as well. So the alveolar surface tension, the thin liquid film that lines each alveolus, display alveolar surface tension. At an air-water interface, the water molecules at the surface are more strongly attracted to other surrounding molecules by means of hydrogen bonds than to the air above the surface. This unequal attraction produces a force known as surface tension at the surface of the liquid. Surface tension has twofold effect. First, the liquid layer resists any force that increase its surface area. That is, it opposes expansion of the alveolus because the surface water molecules oppose being pulled apart. Accordingly, the greater the alveolar surface tension is, the less compliant the lungs are. Second, the liquid surface area tends to shrink as small as possible because the surface water molecules, being preferentially attracted to one another, it tries to get as close as together as possible. Thus, the surface tension of the liquid lining and alveolus tend to reduce alveolus size, squeezing in the air inside. The property, along with the rebound of the stretched elastin fiber, reduce the lung elastic recall back to their inspiratory size when inspiration is over. Pulmonary surfactant is a complex mixture of lipids and proteins secreted by type 2 alveolar cells, as I mentioned earlier. It interbreathes between the water molecules in the fluid lining the alveoli and lowers the alveolar surface tensions. The term surfactant is derived from the surface active agent. Because the cohesive force between the water molecule and adjacent pulmonary surfactant molecules is very low, pulmonary surfactants decrease the extent of hydrogen bonding between molecules at the alveolar air-water interface. By lowering alveolar surface tension, pulmonary surfactants provide two important benefits. It increases pulmonary compliance, reducing the work of inflating the lungs, and to reduce the lung tendency to recoil so that they do not collapse as easy. Pulmonary surfactants' role in reducing the tendency of alveoli to recoil, thereby discouraging alveolar collapse, is important in helping maintain lung stability. The division of the lung into myriad tiny air sacs provide the advantage of tremendously increased surface area for exchange of oxygen and carbon dioxide, but it also presents the problem of maintaining the stability of all these alveoli. Recall that the pressure generated by alveolar surface tension is directed inward, squeezing in on the air in the alveoli. If you visualize the alveoli as spherical bubbles, according to the law of Laplace, the magnitude of the inward directed collapsing pressure is directly proportional to the surface tension and inversely proportional to the radius of the bubble. As we see on the pictures here, it represents the equations of P equal 2T divided by R. B represent inward directed collapsing pressure, T represent surface tension, and R represented radius of bubble. We can substitute T by Y in the pictures as we see on the pictures over here. We can explain more in the next slide as well. So according to the law of the Laplace, the magnitude of the inward directed collapsing pressure is directly proportional to the surface tension and inversely proportional to the radius of the bubble. Because the collapsing pressure is inversely proportional to the radius, the smaller the alveolus, the smaller its radius, the greater its tendency to collapse at a given surface tension. Accordingly, if two alveoli of an equal size but the same surface tensions are connected by the same terminal airway, the small alveoli normally do not collapse and blow up larger alveoli. However, because a pulmonary surfactant reduces the surface tension of the small alveoli more than that of the large alveoli, pulmonary surfactant decreases surface tension to a greater degree in a small alveoli than in larger alveoli because the surfactant's molecules are crowded closer together in the smaller alveoli. The larger an alveolus, the more spread out are its surfactant molecules and the less effect they have in reducing surface tension. The surfactants induce lower surface tension of small alveoli, offset the effect of their smaller radius in determining the inward directed pressure. Therefore, the presence of surfactants causes the collapsing the pressures of a small alveoli to become comparable to that of the larger alveoli and minimize the tendency for small alveoli to collapse and empty their content into large alveoli, as we see in the next slide. Pulmonary surfactants therefore help stabilize the size of the alveoli and help keep them open and available to participate in gas exchange, the opposing force acting on the lung, that is, the forces keeping the alveoli open, and the countering the forces that promote alveolar collapse. So, as summarized in these pictures, is the alveolus, because it generates larger collapsing pressures, tend to collapse and empty its air into larger alveolus. While in this picture we can see that pulmonary surfactants reduce the surface tension of a small alveolus more than that of the larger alveolus. These reductions in surface tension offset the effect of the smaller radius and determine the inward directed pressures. Consequently, the collapsing pressures of the small and larger alveoli are comparable. Therefore, in the presence of pulmonary surfactants, a small alveolus does not collapse and empty its air into the larger alveolus. Let's talk about surfactant's role and function. One of the roles of surfactant is lowering surface tension at the air-liquid interface and thus preventing alveolar collapse at the end expirations. The second one is interacting with subsequent killing of pathogen or preventing their disseminations. The third one is modulating immune response. The destructive change in surface area of alveoli through our respiratory cycle dictate that alveolar surface tension need to be less than 2 nanometer Newton at end expiration to prevent alveolar collapse. This critical function of surfactant is achieved through its maintenance of a film highly enriched in DBPC lipid, which produce extremely low surface tensions, less than 1 meter newtons on compression. These biophysical properties have led to modified exogenous surfactant replacement therapy that have impacted the outcome of neonatal respiratory distress syndrome. Surfactants also play a vital role in host defense against infections. The collecting surfactant protein A and D enhance bacterial and viral clearance. As I briefly mentioned, the C-terminal lectin domains of these proteins preferentially bind non-host oligosaccharide found on viruses and bacteria. The most well-described function of collectin is their ability to opsonize pathogen and facilitate their phagocytosis by cells of the innate immune system, such as macrophage and monocyte, as well as regulate the production of cell-derived mediator. Studies have shown that the mice deficient in surfactant B exhibit impaired clearance against virus, bacterial, and viral infection, including group A Streptococcus, for example, and respiratory syncytial viruses. More recently, surfactants A and D have also been demonstrated to have a direct antibacterial activity against Escherichia coli, and as well as antifungal activity against Histoplasma capsulotum through increasing membrane permeability of the microbe. In humans, there exist two genes, surfactants A1 and surfactant A2, that encode for surfactants A1 and surfactant A2 proteins respectively. This suggests possibility that there may be human subpopulation with differential vulnerability to microbial infection based on these surfactants A isoform. In addition to facilitate and activating the immune system, the lung collectin also act as immunomodulator. Surfactants A can inhibit dendritic cells maturation and inhibit eosinophil release of IL-8, interleukin 8. Also, studies have shown that surfactant A and D inhibit allergen-induced lymphocyte proliferations via multiple mechanisms, and that this effect is blunted in activated lymphocyte from children with asthma. Surfactants A and surfactant Z also bind directly to allergen and particles such as pollen grains, house dust mice allergens, and aspergillus fumigatus allergen, inhibiting a specific interleukin E binding to allergens and subsequently decreasing allergen-induced histamine release. And here are my references. Thank you for listening, and God bless you all.